Hi guys. So after a long time, I am back with another video. My computer has started working again. There was some problem with the laptop webcam, and for these couple of months, primarily because of lack of time. Secondly, everyone was giving me very expensive troubleshoot options. So finally, I got someone who repaired this laptop and made the webcam workable. And as soon as the webcam became working, I am shooting this and recording this video because even I realize it has been very long between my last video and this video. So without any more you know delay, let me start with the second part of karyotyping. I had stopped at fish. So the second part of karyotyping, of molecular pathology, which is karyotyping. Now, karyotyping is the study where you process a sample in such a way that you break the membrane of the cell and analyze the chromosome of the cell. Initially, it was done under a microscope. Yes, you can see chromosomes under light microscope. Excuse me. The cytogenetist has the capability of recognizing which chromosome pair of chromosome is chromosome number 1, which is chromosome number 2, as per the color or the width of the bands which are there on the entire chromosome. So it is possible to see chromosomes under light microscope. Initially, the entire pattern, the set of chromosomes, when you put it on a sheet, the 23 pairs, that thing is known as a karyogram. Initially, it was done purely by clicking pictures on the microscope, then cutting out individual chromosome and pasting it on a chart paper. Now we have softwares, platforms, and cameras to do it for us. But the AI tools cannot replace the cytogeneticist or the morphologist. And I am lucky enough to have been trained for a month in this technique from Spain, and I have seen both the things, the previous method of doing it under a microscope, and I have used the software and I've learned from the best. And I was under one of the most experienced cytogeneticists that I've ever met, Dr. Granada, and she was a wonderful person and gave me a rich hands-on experience for this technique. So the use of karyotyping is to see the chromosome structure as a whole. You can't see the gene inside the chromosome, but you can see the entire structure. Second, you can see the number of the chromosome. Third, you can see if there's any variation in that structure or the shape of the chromosome. Fine. So this is how a chromosome looks: the petit arm, the long arm, and of course then the centromere. So this is a basic of how what a chromosome looks like. We all know, since we all are in the medical field. So main purpose is to locate and visualize changes in the structure and the number. You can break a cell at either the metaphase stage or the interphase. Karyotyping is preferably done in the metaphase stage, and we use chemicals to arrest. Once I get a sample, I culture that sample using RPMI. I get a lot of cells of that sample which was received in the lab. Then I add a colchicine or something which stops the mitosis at metaphase, break open the membrane of the cell, paint the chromosomes, and visualize it under the microscope. This is how karyotyping is done. We all know that males are XY, females are XX, so nothing new. So this is how a karyogram looks, and can you see this blue, and so it is not actually blue and black, it is white and black. So there's this combination of white and black, and we know that this is row A, B, C, D, E. This is A. This is these three are A. This is B. This is row C, D, E. Then F. That way, the cytogeneticists have a way of labeling the karyogram, and see all the acrocentric ones are on one side, the metacentric ones are together. So the telocentric, this is acrocentric. The telocentric ones are on one side. So this is how you actually arrange your chromosomes. Okay. So this I've already told you. Now karyotyping can be symmetric and asymmetric. That is whether you are karyotyping a metacentric chromosome or you are karyotyping a acrocentric chromosome. So symmetric means a metacentric, asymmetric means you have acrocentric, telocentric also. So mostly what we do is an asymmetric karyotype. We read all the chromosomes, not just one group of chromosomes. I told you what symmetric asymmetric is. So GIMSA, there are a lot of stains which you can use for banding. Means to give the chromosome karyogram a color. GIMSA is the most common stain that is used, and what we do is G banding when we are staining the slide. And next lecture, I'll tell you a bit about bandings, but in next lecture I'll take it in detail also. I'm trying to make these lectures, 5 to 7 minutes, not more than that, because it will be overwhelming for you to listen to me for more than 7 to 10 minutes. So quinacrine banding, just like we use GIMSA as a stain, you can also use quinacrine. Quinacrine, mustard or quinacrine dihydrochloride was initial method of chromosome banding where you see fluorescence or UV radiations. On a darker background, the bands would appear like bright fluorescent light. So this is the basic, because why this happens, quinacrine is an intercalating agent with DNA, and the fluorescent compound gives a fluorescence to the band which when exposed to ultraviolet light, which is available in the microscope through which you will visualize this. Okay, this is the most important one done in labs, GIMSA banding, most frequent, non-fluorescent. You require a pre-treatment step. I'll tell you what it is. You use trypsin to digest the membrane, and then we can, stay in the chromosome with GIMSA. A banding pattern, as I showed you in the karyogram before, along the length of the chromosome is created. You need to know that GIMSA stains the AT content darker as compared to GC. So any chromosome where there's more AT, that will have darker bands as compared to chromosomes which have lesser AT. So I will take in detail the bandings next time. Today I just wanted to sensitize to you about what is a karyogram, types of karyogram, symmetric, asymmetric, how to stain a basic of that, and what to look at, the bands is so that what we look at, and why we do karyotyping. The next lecture I'll tell you the stains which we use in karyotyping, and after that I'll show you some examples and cases for this. So stay happy, stay healthy, and I'll see you soon.